Okay, we'll be back, all of you. In this video, we are going to understand regulation of transcription. As we know, of any process, for it to be sustainable, it has to be regulated in some way or the other. And when you look at the different processes, the translation and transcription, the easiest or the most sustainable way or the most convenient way is to regulate the transcription. Because if you think about it, if you control translation, it means you've wasted energy to transcribe a given messenger RNA. So the best thing to do is to regulate the transcription such that you don't have things that you don't need and energy is not wasted. So in this video, we are going to look at it in the broad perspective, and we shall understand how it works and the different examples in which you can explain it. This is your girl Ruth, the .com scientist, here to explain to you the complex biology. Okay, let's get into the regulation. Now, remember we are looking at bacteria specifically. Bacteria have the capacity to adapt to different environmental conditions. And by different, it means the environment keeps changing, and because of the changing environment, bacteria are supposed to adapt to the changing environment. Otherwise, they would be destroyed or their growth would be inhibited. Therefore, these specific conditions will influence which kinds of messenger RNA are formed and which ones are translated to the specific proteins. The level, in the introduction I talked about regulation at the transcriptional level. And we are going to look at the different classes in which transcription regulation is done. The first process is the environmental signals, or those components in the environment can interfere with the transcription. Interfere can be in terms of blocking, it can be in terms of reducing, stopping. So in case the environmental signal is stopping or it's interfering with the transcription, it means that we have negative regulation. Let's get this clear and how the negative regulation comes in. On the other hand, if you have the environmental condition facilitating transcription, it means that it is positive regulation. So facilitate is positive, interfere is negative. That's what we should understand. Okay, now we're going to look at the negative regulation. In the negative regulation, we shall have an example of the lac operon, the lactose operon, or the lac. And in the lac operon, how does it come about? If you have your bacteria in a cell culture and there is glucose in the media, the lac operon will not be necessary. It will be switched off because you have glucose. The bacterial cells are going to prefer glucose. They take it in with ease. Therefore, when the bacterial culture does not have any maltose, it only has glucose, we are going to have the operon switched off. And the operon is more like a group of genes which do a related function, which carry out related function. Now we are going to look at the different genes that we find in this operon, after understanding that it's only switched on when there is no glucose, when there is lactose in the media. We have three major genes in this operon. The first gene is usually abbreviated as B. Sorry, it's the lac Z. It's usually Z. The Z is the gene which contains information for formation of beta galactosidase. And then we have lac Y. The Y is the one which codes for permease enzyme. Then the LAC-A, which codes for trans-acetylase. Now, why these specific enzymes? Lactose does not enter the cells with ease. Therefore, the cell has to become more permeable. It has to increase the pores. It has to increase the permeability of the cell to the lactose. The cell, it means that it will need to be broken down into glucose and galactose. If you remember, glucose and galactose give us lactose. So the enzyme which breaks down the lactose is beta-galactosidase. However, the role of LAC-A, or the one which codes for trans-acetylase, is not known. Good morning. Is not known, therefore, that one we are not, but the basic two are LAC-Z and LAC-Y. And we have to keep in mind that when there is no lactose, these genes are not transcribed, they are switched off. They are switched off. So we are going to describe this a bit. Now, as I said, when there is no lactose in the media, we have an active repressor. That repressor is going to bind close to the promoter. When it binds to the promoter, RNA polymerase is incapable of binding to the promoter for it to continue transcription. Therefore, it is going to inhibit. No, that is when that is how the negative regulation comes in. Presence of that repressor protein on the next to the promoter site is going to inhibit the transcription of the gene. However, when lactose is present, that repressor molecule is going to leave that site, and lactose is going to be able to bind. Is going to be able to bind to the RNA polymerase is going to be able to bind to the promoter site, thus initiating and continuing with the transcription process, thus formation of these different enzymes which are going to be relevant to the metabolism of the lactose in the media. So that is basically how the lactose operon operates. We have to put emphasis on those different enzymes and the different genes which code for the enzymes, such that we understand how the negative regulation comes in. We are going to look at the other example in the positive regulation, and that is going to be the maltose operon. Now, with the maltose operon, here there is regulation or the control of expression of the maltose operon. We are going to look at sorry for that noise. We are next. We are very close to the airport, therefore the aeroplanes just fly so close to us. Okay, I hope you're with me. The active gene product binds near the promoter of the genes involved in maltose transport and catabolism. So when there is binding of this active gene product, we are going to have transcription taking place. So that is how the positive regulation comes in. Now in this picture, as you can see clearly, we are seeing that DNA, which has the genes coding for the MALT-T, is going to code for TI. The TI is the inactive gene product from this specific sequence. Now, when that gene product is formed, when there is no maltose, it's going to remain inactive. Therefore, it's not going to be able to do its role. Now, when there is maltose in the media, maltose is going to bind to that inactive gene product, and the moment it binds, it's going to form a positive. It's going to become positive. It's going to become activated. Its activation means it's able to bind to the promoter side, promoter part of the operon, and the moment it binds,
transcription is going to take place. So that is, you have to take note of those two. Binding of that gene product activates, or it allows for transcription to take place. However, in the lactose operon, when there is that protein binding, it's going to interfere with the transcription. So basically, that is how gene regulation is done. And if you have any questions, please leave them in the comment section. I will try to answer them as soon as possible. And then in the next video, we are going to look at how translation occurs. We've now made our messenger RNA, we've regulated it. Now we are going to look at how we are now going to make the final product. Thank you so much for watching. I hope you learned something. I hope this can help you remember things. You can subscribe to the channel if you're interested in more of this content, and that bell, just hit on it so that you can be notified whenever I post. Thank you so much. I'm going to for these kinds of videos, I'm going to try as much as possible to look for the best animated video, such that you can really visualize, you can see what we are talking about, for it to be able to stick into the brain and never to leave. Thank you so much, guys. I wish you all the best in your endeavors. Bye-bye.